Good everybody. So I welcome you to this seminar, which will be given by Arthur Villordon, who is a professor in the Louisiana State University Agriculture Center's Sweetpotato Research Station located in Chase, Louisiana, in the United States. Perhaps we should introduce ourselves one by one, and do so. I would like that you use the microphone, and then use Professor Villordon to all of you. My name is Marc Guillard. I'm a plant molecular biologist and the head of one of the 6 research programs of SIP on game-changing solutions.
I'm James, a research associate with the International Potato Center, working on potatoes. I'm specializing in plant agronomy, so majoring in plant nutrition.
I'm Benjamin Kivuva. I work with the Kenya Agricultural Livestock Rights Organization. I also do some breeder.
Good afternoon, everybody. My name is Brahma Lonjala from SIP. I'm a research associate based at the Kenya Plant Health Inspection Services in Buga. I work on, I do research and also manage the Sweetpotato Jam Plaza in Kenya.
Good afternoon. I'm Marshall Tegaraman. I'm the head of the Sweetpotato for Africa program that's achieving objective 3 of SIP.
And good afternoon. I'm George Abong from the University of Nairobi, Department of Food Science, Nutrition and Technology. Do research, also lecture. Thank you.
Hi, once again, I'm Tawanda Muzingi. I'm a food scientist, STEP, and I'm looking forward to your presentation. Thank you.
Good afternoon. I'm Webex. I'm research associate with SIP, working on developing potatoes resistant to bacterial yeast.
My name is Eric Makembe. I'm a molecular biologist working on potato and trying to develop varieties that are resistant to leaf blight and bacterial yeast.
Good afternoon, everyone. I'm Immaculate Makoko. I'm a research assistant working on potato yeast. Good afternoon, everybody. Margaret McEwen, working on sweet potato seed systems.
Good afternoon. I'm Binata Mongo. I'm a junior research assistant, and I work for Potato Resistance Project.
Good afternoon. I'm Dr. Clemente. I work with the genomic tools for sweet potato improvement. I'm a research technician.
I have also online Mercy Kitavi. She's a postdoc in molecular genetics of the sweet potato, working with the genomic tool for sweet potato improvement project led by NC State University. I'm sure you're aware of. So, sorry.
Because I'm a regional communication specialist for SIP Sub-Saharan Africa.
Sorry for that. So let me introduce Arthur to all of us. I understand that you are actually coming from the Philippines originally. You graduated from plant breeding from the Visayas State University, and an MS plant breeding from the University of the Philippines in Los Baños, and a PhD in plant breeding from Louisiana State University. So my research has addressed morphological and genetic variability of sweet potato, And your research interests concern the roles and interactions of environmental management and intrinsic variables that control root architecture development, store root formation, and shape determination in sweet potato. I think let me ask you to start, entitled The Role of Phosphorus and Soil Compaction in Determining Sweet Potato Storage Root Length and Shape. Thank you very much, Arthur, for coming and who gives this lecture.
Thank you, Margaret. Just introduction. Thank you, Margaret, for the hospitality and organizing the program here. Just came Ethiopia first 5 days last week, and as part of a Berlog CGIR fellowship program. Then Jan kindly invited me to visit by here because I'm already here, and so very much enjoying my visit and learning a lot. So, okay. As I back this presentation, about 2 years ago, a French fry company, we visited with them, and they indicated that they prefer sweet potato with a certain shape, that are tapered ends to that recovery. So obviously, I have realized illustration here showing a sweet tapered end and a blocky sweet potato. Okay. One of the reasons they prefer a certain shape is that during processing, the sweet potatoes are using a water knife at a very high velocity and higher than potato, because potato is softer than the sweet potato. So they require to be able to do this, to be able to achieve the consistent slicing. So you can see they probably lose up to 30% of the raw material just fall floor, scattering about because of the desired shape, not what they need it to be. If they had their own way, they want a sweet potato to be a brick, so that all of the product are 100%. Because to them, if they lose 20%, 30%, that's lost profit already. Okay, so it's an incentive for them to be able to do that. So we've already done prior work on this, but have come in, the industry come in and really express their need for that particular direction is an incentive for us in research to try and really focus more on the subject. It's a challenging concept. Can we control sweet potato shape and length? So basically, the research objectives are first to understand the external, meaning environmental management, or can be agroclimatic, and intrinsic genetics determines shape determination, and then based on that to develop and test management approaches. After all, the French fry company came to us because they want the growers to be able to adopt what we find out, mean something in the management process. So to them, it's not only the science of it, but for more practical ends, with the growers to follow standard management procedures so that when they bring in their product to the plant, they'll be able to refer their product efficiently. We really don't know much about sweet potato. Breeding programs are trying to for a desirable shape, but in terms of the science of it, there's not a whole lot. And what I thought, I said, okay, I have a fresh start here. What if I look at the model systems? And let's say Arabidopsis. No, it's a long jump, but let's just say, okay, what if we look at Arabidopsis and then we look at what controls its root architecture? And in fact, its literature, there's quite a bit of work of it. And I like this paper in particular because it sums up, is known in Arabidopsis in terms of what controls root length. And then points out that phosphorus is actually one of the determining variables in root length determination. For instance, in their work, they're saying that high phosphorus is actually for the root to grow long, meaning in whatever medium they grow the Arabidopsis plant, the roots will continue to grow as long as phosphorus is there. On the other hand, phosphorus is deficient or low, it's a signal to the root to stop growing at some point. And so the red there represents the meristem. If phosphorus is available, the meristem is active, the root will grow. If the phosphation or not present in the medium, then it's a signal to stop growing, and in fact have a shorter root. So evidence in terms is that the phosphorus stands at the root tip. That's very profound. Now, if you can translate it, though, or even other root crops, that's even more, the potential science that can be derived from that concept is very, very interesting. So, before, actually look at this, because I work with our growers directly, and phosphorus is an input. Farmers phosphorus to apply in our fields, and sometimes they apply so much, sometimes not. So there's really a lot of things that can happen here. First, we have to be able to generate basic information, and then the applied aspect of it is direct. We can almost say that to a grower that you need to apply this amount of phosphorus to be able to achieve yields as well as the desired shape. So this was an attempt at translating the findings in Arabidopsis to sweet potato. So we have two experimental treatments, one variety. The left image shows a variety which you call BioBell. It's sweet potato processing. As you'll notice, it's red skin. Right? In the United States, the preferred skin color is not red. So a store there in general, you go to a store there, you'll not be able to see red skin sweet potatoes unless it's a specialty store. It's going to be a copper skin, but the fry will not care what skin color it is because you're only after the flesh. But in here, you'll see that two treatments with 7 kilograms per hectare of phosphate, and on the right panel you see without. Now, it is important to state that, go back, that the soil here, by analysis, had already 35 ppm. So, starting with a zero phosphorus in the system. And that's the disadvantage of field work in terms of phosphate, no phosphate, because in the field you're going to have some phosphate available. But do that if you just take out the phosphorus from your management program, you're able to change one thing, and that is some circularity. Whereas here, that is paper dense, which the industry would not like to see, and here they're losing paper dense. Not completely. As you can see, the effect is measurable. Now, shape. Here to study shape, you have to be able to measure it. And right now, our conventional way of measuring shape is using length and width ratio. Okay, so this sweet potato on the left side obviously is round, and so the length to width ratio is more than the length width ratio of this circular sweet potato. As that is one, then it is circular. However, still a dimensional shape metric. We're not measuring the actual shape here. We're just measuring the two dimensions of the object. A more relevant metric would be the surface area to volume ratio. If you look at this angle, if you try to achieve if you try to shape metric, then our goal is minimize surface area to volume ratio. Now, the phase of post-harvest is that if you have a lot of surface area, then you have a lot of area to lose water from. You minimize the area, maximize the volume, then you not only conserve the product longer, but you also minimize damage during harvesting. The papers tend to lose, its tips will tend to lose a lot of tissue because we do mechanical harvesting. And so if we harvesting with all going on, post-harvest life will be shortened. In other words, if we use surface to volume ratio, we are targeting two things. First, we maximize product recovery, at the same time maximizing post-harvest. So basically, the hypothesis was that we think phosphorus is going to be involved in root shape determination in sweet potato. And understanding what makes shape it is will lead to the development and testing of management interventions. Okay, so as I suggested, there are a lot of challenges of measuring root length in the field, and we need to really develop a good system to be able to do that. If our treatment is phosphorus, that means everything should be perfect. So I'm just going to give some of the variables that we would be addressed to be able to really measure phosphorus effects. So the first thing that we were concerned about is that our seed needs to be uniform, and our vines are going to be uniform. So if some of you are familiar with the production cycle in the United States, in April, we take out our stored sweet potato seed, our roots, and bed them out. Now, that's a very small bed. Okay, it's a bed of our seed beds. Commercial growers probably have 2 hectares of seed beds. So we do this around April, around Easter, and by June we will have plant beds looking like this. And that's what we use for our propagation. That's where we start. In order to there's uniformity in the vines, I tend to do the cutting myself. And sometimes family would show up. And that's my wife and my son helping me cut vines. Okay. And so this is the length of the cuttings or vines we use. It's about 30 centimeters. And you will have roughly 2 or 3 nodes. And the variety in general would be varying about 2 or 3 nodes. Another variety to address is uniform planting depth. Obviously, vines are set deep to the soil, therefore you'll expect your roots to be shorter. If your vines set higher in the soil or are near the surface, then you'll have longer roots. Also, if you plant vine vertically, some nodes are higher, some nodes are lower. Okay? To address that possible variable, we plant our vines flat, horizontally, just like what's shown here. So that whole root zone is going to be exposed on we minimize variation. We also expose nodes to uniform growth potential. So if hard sand here or a area, then they'll be exposed to the growth pattern or limitation. So as you can see, this is one hill of this would be variety Orleans. So it's setting about 8 cents, more or less of the same length, and that's about 50 days of sampling. Soil moisture, very critical. Our farmers typically use center pivot irrigation or flood irrigation. That can be varied because center pivot overhead can be subjected to wind and that's the uniformity. So we decided to use trickle irrigation or drip tape. So and we also instead of putting the drip tape on top of the row, we buried the drip tape in the root zone. So the water is right there in the root zone, and hope that it will reduce the variability. So the first panel shows some of our. We had several planting dates. Okay, so we start to plant in May, and then all the way to the middle of June. So we had several plants to be able to address that variable of different planting times. May, we still have cooler nights, 24, 25 at night, and then we'll finish up in June. Your nights might be at around 27, 28. So we try to address that variability as well. So the middle panel will show the crop at about 60 days, and we typically are starting at 90 days for some of the trials. So This is our plot digger, and as you can see, the drip tape goes on the chain and falls up behind it. So it's really to manage the drip tape if it's buried. Okay, flat. First panel shows some roots at 30 days. Very nice uniform development. And this is Orleans on the second panel. It shows planted on May 11th and sampled 30. And there's a scaler so you can see how much it's doing after about 35 days since planting. So again, some nice uniform length roots. So you have this system, and you have one treatment showing that the root is shorter than the others. Then we know that we've reduced some of the variation, and you can say that it's the phosphorus that's affecting the not other variables. That's the theory. Youth planting is a recent phenomenon, or it's an older phenomenon, but it's actually being used all over the world. So this is, I was in Australia in 2004. I'm sorry. Yes. Up, down. But I thought we didn't test right. So anyway, that's just funny. I'm sure it was the other way around last night. But in Australia, do everything by hand. And dig a trench, as you saw, and lay line flat. And so this is one row. There were 20. Yes. Oh, that's yes. Everything's opposite. That's so funny. Yeah. But last week I mentioned that I was in Ethiopia, and well, let's see the potential in Australia. It's amazing. You can engineer the vine. You can engineer the environment, and as you can see, there's not a single root that did not initiate. I count 8 to 10 storage roots per hill using that approach. The one thing that is that there's also tape nearby. When they're planting, the drip tape starts. Where the drip tape says there's water, so that water is available from the get-go. And if you see the growers there, they will insist that if you don't water a plant, you will lose half of your yield. So I mentioned that I was in Ethiopia last week, and so we went to a farm, Margaret, it'd be East Hawassa, right? Yes, okay. And so this farmer is among 5 or 10 farmers in this village. They have a co-op of this, and this would be a white flesh variety. He also had a plot of orange flesh sweet potatoes. Him, how do you plant your sweet potatoes? So he got tool, and then he made a trench on the soil and lined flat. And then he said this is about 2 or 3 months. So, but that he said that he discovered that it gave him a better yield compared to his traditional 45-degree planting. I learned a lot from you today. We thought everything accounted for that. Okay, we uniform water, uniform cuttings, consistent seed. But there was one thing that confounded us, and this time, if you notice, this particular, okay, so it's flat, right? You bend the vine right here. So look at this root. It's a trait that is now challenged because that is shooting to the side. Shooting to the side means you have this ridge that you're growing in. It's going to be longer. It won't follow your direction of going down. So, but this appears to be variety specific. This is Beauregard. And so this is something, a trait that can be selected for. Because your root's shooting down, And if you're a breeder, and if you have consistent roots, and you have some, going this way, then, it's probably something that you can select against. The other that you should notice is that this root is going straight towards the drip line. The drip line is here. So maybe this particular variety just wants to get where the water is, and it's trying to follow where the water is, obviously. So may not be a good trait for the French fry company. So treatment plan was simple. Some plots got 0 phosphorus. Others plot got 67 kilograms per hectare, 134, 201. Now the other inputs are nitrogen. We put about 50 kilograms per hectare. Potassium, we put about 150 kilograms per hectare. Our pH is about 5.2. Boron was sufficient, so we didn't put that boron. I'm saying this because that's what typically our growers do. They soil analysis, and if the boron is low, they apply boron. If sulfur is low, they apply sulfur. So, if you notice the variety BioBell, E means no phosphate applied. And yield is 50 pounds per acre, but there's a conversion here. So, tank would be 25 tons. This would be this variety very high yielding, so the fry company likes it, even though it's not sold in the fresh market. So it's bred specifically for processing. As you see, meaning no phosphate was applied, the yield was higher when treatments where phosphate was applied. So that phosphate is something that you can really manage, because even if you don't put phosphate out, depending on what your soil status is, in case the soil showed 34 ppm of phosphorus. So Sweetpotato requires very low phosphorus. And if you soil it, there's no need to add phosphate to your fertilizer. And that information is applicable to our growers there. We can tell them based on your soil analysis, you don't need to put out phosphorus. And sometimes they put out 120 grams just because their consultant told them to do it. So the consultant, to do that, they pay for fertilizer to reach their yield. The numbers don't add up. Okay, so trial we did in 2015, and I only had 2 levels of phosphate. You have 67 kilograms to your left, the checks, and then 134 kilograms. This time we used 5 varieties. So 8 is a variety for different phosphate treatments. The one thing that is that there's a varietal response to phosphorus requirement. Some varieties low phosphorus, others not. These varieties, imagine the diversity of the material probably out there to be able to select for phosphorus efficient variety clones. So this BioBell is a planting variety, and yields are tremendous, over 50 tons per hectare, even without phosphorus. But some varieties are low, and to autonomous varieties require some phosphorus supply. So as you can see, that's on some of what additions of phosphorus. The gaming has really spurred the development of sensors, including 3D sensors. And this is just a very inexpensive sensor that attaches to an iPad. The picture here doesn't show the sensor, but it shows me, my hand, holding the iPad and scanning the root. The root is here. It's being suspended using a wire. That's the only thing I can see. But you can the root within 2 minutes acquire the 3D shape and the solute, the surface area, and the volume of the sweet potato. So if you have so many samples, then acquire 2 sensors. But this was just an initial development work. But as to show that it is now feasible to be able to acquire root shape, surface area and volume. And currently how this is being acquired, the volume of the root and the surface area. Can anybody tell me that? The volume sounds easy. You just put it in a beaker and you measure the distance, right? Now for surface, how do you think it is being measured? Well, one way is to put cover the root and then take it out and scan it. Use a air scanner or a scanner. That's tedious. Okay, samples to process. It will take you forever to do that. But the scan enables you to acquire those parameters. So, the left panel shows the same thing, Orleans. One came from a phosphorus treated plot, and the bottom shows some source obtained from a low phosphorus plot. So you can see that sweet potatoes are circular, around. And then looking at surface area to volume ratio, what we are looking is we want to surface area. The concept is reduce water loss in storage, as well as maximize the volume, maximize the yield. We heard that how the desirable shape is this for processing. But if you go here, that means very high surface area, and the loss is great in storage. Now, if you go down here, it means your root is small, and you don't want all roots. So there is an optimum surface area and volume that shows you what shape that is. In terms of reading, it will show you what your idiotype would be in terms of shape, what the traits are. I mentioned earlier, phosphorus is not the only variable, and in that, we've seen that root angle can confound your result, but that depends on variety. So, halfway through this, I was trying to scratch my head. My roots are not behaving like they ought to be. I came across this very old reference, 1896. In reference, Mr. Fry wrote that to obtain well-rounded tubers, sweet potato, which market in 1896, the ground did not be broken over 3 or 4 inches deep. That's about 10 centimeters deep. And now, if you want to control your shape, don't get ground deeper than 12 or 15 centimeters. He observed that in 1896. It's time to, to verify that is. So essentially, Mr. Pratt, okay, so if I can put my vines in a pot and I control the depth of the growing zone, it can dictate the shape of the sweet potato. So now if we can grow them in containers, it will look like this. So the container on the light has, on the left has a hard pan. It's a barrier. So if you grow the cuttings, they can grow until they hit the barrier. Now on the other side, there's no barrier. And this is now that for as long as the root goes down and doesn't hit the barrier, it will not swell. The moment barrier, there's nowhere else for it to grow, but it starts swelling. In effect, you are designed to start swelling the roots and those plants planted in a deeper soil where there's no barrier. And that we're able to determine rising, potentially the earliness. So that's the principle, that's the theory. Okay, so we design tests in the greenhouse where we can control pretty much everything. And the existence of a hard pan, we just sand and just press it down and measure the pressure. And so it's been determined that roots cannot penetrate 300 psi. I have to find the conversion for that. But at that density, roots cannot penetrate. So we are able to measure the density. So it's something that we can quantify. And the other half of the treatments, we just pour the sand. So if you put something that measures force, them in this growth state and not encounter any resistance. Okay? What we got? Again, the variety at 20 days. So, obviously, the root system on the right was grown that part with the hard pan. And as you can see, even the system, I mean, the feeder, the back roots here, so with the test include phosphorus now. So we combine phosphorus and hard pan. Okay, so roots from 4 different replications. One part is one replicate, and this is with phosphorus, no hard pan. So it's loose sand with phosphorus. After 50 days, some of the roots are already about 12 centimeters long. Okay. The top, what you saw before, the bottom panel is plants with hard pan. As you can see, induced decay and the diameter is only slightly larger than the diameter of the other. So it helps to stop growing by barrier. It's not for them to start swelling outside. And it's 50 days. If you grow this for 50 days more, you'll end up with brown sweet potatoes. So we've added a third condition here. So this was the check. With phosphorus, no hard pan. This is phosphorus with hard pan. Without phosphorus, no hard pan. If you notice, the length of the storage root is about the same with phosphorus. Now, phosphorus is also playing its role. There's no hard pan. It should grow long, right? Because that has phosphorus, which is as strong as the one with hard pan. Now, it acts as a vertical hard pan. Don't add phosphorus. So these are measurements from the test. That check with phosphorus. You add the hard pan, you effectively cut the length of the storage by half. Phosphorus, the length is the same. That's bibell, and on the other side here you have some genetic funds there. So are able to respond better to the presence of phosphorus, others not. Again, opportunity to be able to select for that particular trait. My technician said, why don't we design a test where we have very deep soil? So I came up with this. Okay, if you notice, they're all the same height here, but you know, this chain is about double the length of the others. Everything seems glowing here because we use LED lights. This was done in winter, where our days are shorter, so we put in our lighting using LED, and it looks like it's red. In fact, it's red. It's red at night. And this is what we got. So I don't need to see which roots were grown in the deeper pots. And as you can see, this particular plant is expending energy to grow downwards. Instead of storing that energy in the storage root, it's using it to grow downwards. Well, this is already starting to store its energy into storage roots. So that the root system out resources available at that time, and until it stops finding that resources to continue to store it. But when it's available and stops growing downwards, starts to swell. You can actually manipulate that. So in conditions, we cannot control hard pan. In conditions, there are soils which are heavy, like soils, and in some cases it is sandier soils. How would you describe these soils here in Kenya? The sandy loam, very variable, right? So it's a challenge. Okay. Variable to consider. But in this particular test, we wanted to simulate light soils, meaning sand, and heavier soils. What we did is add more sand to the pot, shake it, and measure the resistance. So we'll be half a kilogram more to this pot and shake it. So we know it's consistent, and then we measure the resistance, and then we have that. And so we can measure this. So the idea is we were going to screen varieties to see if a variety is able to penetrate that force better than others. So we wanted to find out if there's a varietal effect. Force you hard, so we always use it as sec because it is grown all over the world, pretty much. So this is 50 days. So you have 4 reps for compacted on the left side and not compacted on the right side. And as you can see, the roots tends to be longer, and it's not okay. But the other thing that you'll probably see is that the portion of the lateral roots here looks more than the proportion of the roots over there. So if compacted, we know that they stop growing earlier than the others. They start swelling. As you can see, this was essentially trying to store or trying to take up water instead of stop growing, start swelling roots. This is another variety that we grow, pronounced storage roots of 50 days. Again, the difference in the total root mass and the variety. This is another thing that we talking about root length here, but in general, bells to respond slightly different in terms of compact. We think it actually affected its root development. So there's another observation that we thought that was significant is that some varieties may not like compact soils at all. In other words, it can actually press their yield instead of encouraging it. And then variety. So again, the same funds as Bellevue. Non-compact soils, advance indeed better. The yield potential is better. But looking at the compacted soil, it's reduced by almost half. So this is something that we're not looking for, but we thought was meaningful because it will tell you that maybe the grower needs to till the soil if he wants to grow evangeline, not leave it compact. So this is just from the prior test, and as you can see, quite a range of variable response among those varieties used. So these are the varieties grown in non-soil. Evangeline had longer roots. Providence and North Carolina variety was shorter. This would tell you that some varieties in certain environment soils, many varieties which are heavier soils because they are adapted to that particular soil. Variety looks like it's long in one environment, intensive in another. It's very sensitive to the soil pressure or compactness of the soil. And those are grown in the compacted soils in the lab. So any have soil that's good or density, it will really affect your yield potential. So, understand the phosphorus. So we know that phosphorus works with soil component to be able to determine how long your roots are in the shape. But I want to isolate compaction. I want to really understand the role of phosphorus only in length, but storage is for me. So I designed an experiment where I purposely stop phosphorus at 10, 20, 30, 40, 50 days. So these were the roots from pots with phosphorus for 50 days. Okay, so you have the scale there. Another treatment phosphorus only until 40 days and 30 days. Okay, so if you'll notice, if I do phosphorus all 50 days, it's longer than where you stop phosphorus at 30 days. If you stop for 20 days, you'll phosphorus the length. Finally, this will show you the whole range of response. You know, if you add phosphorus until 50 days, there's no phosphorus at all. So plant grew for 50 days without phosphorus. The roots around. And that's the same variety. And we only change the phosphorus. You know, potassium has a role in soil formation, but phosphorus has a very role as well. If you don't have them, you can have round roots. And you can almost, you know, to the day stop the phosphorus. In other words, fundamentally, phosphorus is required for storage root formation. We know that phosphorus is an important component of nucleic acids, without which you don't have synthesis of cell material for your storage group. So it really makes sense. Looking at agriculture and how phosphorus applied, this is a very slide that a colleague of mine, Dr. Diaz from Kansas State, provided. This is in corn. That's corn, but this was trying to monitor what happens to phosphorus in agricultural soils. Okay, so you sort of can see where green portion of the graphic has high phosphorus content, and if you go red zones, the phosphorus is lower. Phosphorus nutrients that stays in the soil and doesn't move around. And your roots can't get to them, but can take them up. It's unlike nitrogen, which moves around, so the roots can be here, but nitrogen can sort of move to the soil so it encounters it. But phosphorus slides directly to the root zone. So as you can see, if the phosphorus is broadcast, so take your applicator and broadcast it. If you do, you're wasting a lot of your phosphorus because the root zone is in that row where the root system can't get to it. So, and this was to applying the phosphate input in the row where the zone can do it. In other words, in terms of efficiency, phosphate in the roots, abandoning it, increase recovery of the phosphate, you reduce environmental damage, and you maximize plant growth. And so recommending to the growers, and in light of what we know, what we recommend to the growers is that you apply the phosphate, band it in the row, and not broadcast. So, management agents. This study is all about understanding root shape and eventually trying to apply management strategies. So, for cultivation necessary, what variety is grown. For the soil, compaction might be good, but for others, not, as we saw. So, this is where knowledge of the variety characteristics come in, because you can modify your environment and be able to do what's required. For luscious soils like sand, some roots will be long. About it. Maybe apply it only in the top 20 meters where you want your root zone to be, if it can be helped. Also, maybe watering it so that you don't overwater. Some varieties are sensitive to heavier soils than others. In terms of applying the phosphate inputs, it needs to be applied to the root zone. We've done some experiments to understand what genes are involved. Earlier that there are no genes for root shape. So what genes are involved? We know that phosphorus is involved, right? And so we need to look at what genes are sensitive to phosphorus signaling in the root system. And it's this well worked out. They have a very good understanding of the signaling pathways, transcription is involved. And we're starting from scratch. We've already translated the role of phosphorus in root length, so why not translate, some of the candidates? So we have genes that are activated when phosphorus is low, and they phosphorus starvation response genes. They are genes that respond to that environment. But when phosphorus is active, there's also are responsible for transporting phosphorus. It's one of the nutrients that require an active transporter. Boron requires just water flow, but phosphorus is one of those nutrients that require an active transporter. Transcription factors. We have a working here that's known to be very active in stress type responses. So it's been documented to be involved in phosphorus sensing as well. So we are keen to see if this also active in Sweetpotato. So we did RNA and DNA extractions trying to find out which genes are active. We did blast search, and sure enough, it checked as an Ipomoea nil, probable working transcription factor. And on the bottom here, it shows the phosphate starvation response gene in Ipomoea nil. And so those reference genomes are very useful. This is just genomic DNA. We're using the primers based on those on Sweetpotato. So we had some of those PCR product sequence, and this is sequence information from 4 different varieties. If you know the bottom variety, probably possible deletions or insertions. And we did some analysis, searching for a standard method to be able to tell the grower this variety requires less phosphorus, this one requires more. I think we have a very nice system where we can classify varieties based on phosphorus requirements. I think a nice way of really using molecular tools to be able to support agronomic work and breeding work. And so I need to add more varieties, of course, but this proof of concept that, when you use some of those candidate genes, able to really identify genetic basis of your. So in other words, if you talk about what genes determine root length, we talk phosphorus sensing genes. And that's all I have.
Thank you very much for this presentation. I think the floor is open to questions. Maybe what I can do is I can read the question from Mercy. She's in, forgot to say that she's in Michigan State, in East Lansing. And so maybe she can react to the response while we go over the group here. So her question is about the genetic basis of root shape. And what she's asking is whether anything is known about what is the epigenetic component of root shape. If you become that in Arabidopsis, when you do DNA methylation, they change in transposable elements, which may lead to various changes. So, in particular, morphological abnormalities in roots. So, is there anything known in the case of Sweetpotato about the epigenetic component of root shape?
That's a question, and in fact, you can validate some of these phosphate-acting genes. That's really a springboard for exploring methylation, epigenetic changes, but right now, virtually none about it. But that's a very good question.
Okay, so Mercy, you have a lot of work ahead of you. If you want to investigate, you're welcome. Any question from here in the room? Emma, please.
instead of potatoes are irrigated, huh? Here, most of the potatoes are not irrigated. In that respect, we have very often water deficiencies and goes down into deeper zones. Therefore, longer roots must be desirable to have also, and we have to have a different concept here and different. So we need to balance it out. What suits best, and maybe phosphorus is very, very true to water uptake. On the other hand, when water is going deeper, and when there's a kind of dry season approach.
It's a good point. There are other advantages of deeper roots. Is you're also reducing possible weevil damage, and so that's a very good question. And I just don't know how to answer that. But we also know that okay, we understand that phosphorus is a must. It needs to be there before swell. Now, the thing is that phosphorus is only found in the top, very top soil. So maybe if we're looking for deeper roots, we have to find out if there's a trait that can will let you find those phosphorus from deeper part of this and channel it to the formation zone. Because if you have the premise that phosphorus is needed local sensing, not universal sensing, then where there's no phosphorus, then there's no swelling. So we need to have deeper, cheaper roots so that it will acquire water during times of drought. But we also to be able to have storage roots. And know where phosphorus is, then that's something that needs to be investigated. So I'm trying to understand what the phosphorus status is in the soils here in sub-Saharan Africa. Yes, very good. Yeah.
Other questions?
My concern the changes in the phosphorus content, how they affect the metabolites tubers. Have you looked to see, does it change other like vitamin C, iron, zinc, whatever other components?
That's also an area that we haven't really looked at. We've only looked at basic dry matter content and appear to change it. But it's quite possible that any variation in phosphorus can affect other metabolites as well. So, for example, we look at storage root formation. If phosphorus isn't there, it's not forming roots. So it's a basic need for the root to be able to swell. So it affects any downstream metabolite activity. So, but in looking at the different we added, you notice that if you add 20 kilograms, the yields will go down. So there's things going on that if you put phosphorus in, that you actually reduce your yield. And that's that really needs to be looked at.
Maybe a comment, maybe you can also respond. In relation to the root shape and the amount of phosphorus in the soil, in experiment there is whereby you put 0 phosphorus.
Mm-hmm.
The soil should add some amount of phosphorus. Some at 2.
Yes.
That the plant still needs phosphorus. But now, to determine the kind of shape, and you need to control the amount of phosphorus.
Mm-hmm.
Maybe the question will be, what is the minimum amount of phosphorus which is in the soil? And then it's required shape.
15 ppm?
And that was my question.
Yes.
Concerns that amount to add in those do different varieties require this amount of water?
that question. The short answer, yes. Different varieties require different water. For example, in 2008 and 2009, we had 2 hurricanes, and there was rains for 3 days, and Beauregard in the field. If you have nowhere else for the water to go, it was there, and your roots become asphyxiated. But in Evangeline, the farmer was able to harvest. That goes to show Beauregard probably requires less water than Evangeline shows in many areas where Evangeline is grown. This one particular farm in Honduras applies much water that they say only Evangeline is good for our environment, and Beauregard, no. But he's not aware of the fact that there's varietal differences in terms of water needs. But yes, it's something to do with their capacity to use water. There's probably some other basis for that. Petrol that we don't know of, but I think that it has something to do with how much water is available in the soil, how much water can the plant take up, and the transpiration. So if being not adapted for environment, then that particular variety won't do well. So in other words, some varieties are going to do well in areas where there's less rainfall because they're adapted to that, and it goes back to varietal differences.
Mine is in sub-Saharan Africa, few farmers use fertilizer, and I've hardly seen any rounded sweet potato with 0 phosphorus added. So could you say the soil architecture, or could it be that we don't get that kind of shape?
Okay, are they varieties or orange flesh varieties?
The white.
That's a very good question. I observed that. When I was in Ethiopia, the farmer showed me the white flesh, they were long. The orange flesh was round. If I were to answer then what we know, the hypothesis is that the white requires lower phosphorus than orange flesh. The other is that the white flesh trees are able to make the soil microcommunities that enable to take up whatever phosphorus in the soil. I didn't present data, but in apparently, if the phosphorus is low, the root system exudates. And this exudates dictate what microbes are active in the soil. And some of this what you call as microhazate. And they form a root system, and they help the root system more phosphorus than what it can with its existing root system. That because these varieties, these clones, these landraces have been grown here for years, that they're able to adapt to that environment. But that's just, but because I've seen that the varieties tend to be longer than the orange flesh. So something that needs to be looked into.
Thank you for the good and informative presentation. And mine is just maybe to confirm something for the methods of extraction for determining the available phosphorus. Mostly we use the merit.
Yes.
One, mostly use one, but
okay.
Three still okay. So maybe for comparisons and so on. And as Elma mentioned, we love very acidic soils,
yes.
And the pH is low, so the availability of these in Kenya is a big problem. So I just wanted to confirm the method of extraction for the nutrients.
We use M3, just because our soils are about 5. I like it that way. Yes.
To comment that this research was brought about by the need from private sector,
from industry, yes.
from the processing guys hiring a particular site, what can, as a public research institution, to kind of forge type of interaction organization so that we can do research that is more relevant to the end user?
That's a very good. Ultimately, we want to serve our industry, and they have an expressed need to be able to do something to improve their profits. And they're early on in the planning of the experiment and outlining of the needs. And I think it's a very good way of approaching it, instead of, giving them command of the process. But really works because you are certain that what you're doing is something that the industry needs. And for one of our deliverables was to develop a playbook. So we're to write out our recommendations, and they will use this playbook to give to their growers. So the fish fry industry has contract growers and also non-contract growers, but the growers are supposed to follow their playbook. Part of my work is to write it out so that they will try and follow that in order to produce product that's compatible with the systems. But I think public-private partnership in the long run is important, because it will really not only make your work relevant, but you'll also obtain funding to do so.
That through private?
They to do work.
May I ask something, or after?
Sure. So mine is in relation to our farmer practices. So like in most African countries, we know that farmers do mounds or ridges, and they believe that the larger the mound, the larger the tuber. So what would be practical in terms of having a situation you have a uniform depth in terms of soil compaction? What would be the practical scenario in a real setup, especially considering that in most of our farmers don't do mechanized agriculture?
Yeah, my question is about aeroponics, whether you have tried this for research purpose, and how does Sweet tell us a bit more about aeroponics, if you have
Shown some of the. Okay, so aeroponics, it's not hydroponics, but the principle is that you use misting to be able to roots in a container. So the mist contains nutrients. Now, if you use sweet potato, the roots will not swell because underwater, and we know it's underwater, it will not. So the aeroponic system is a very good tool to understand what nutrients make swelling roots to swell. Now, we've either successfully developed a system where we can make the roots swell in 30 to 35 days just by controlling the nutrient that in the mist. So, for example, we use half-strength Hoagland's to spray it, and the roots swell in 25 days. We tested the phosphorus because, all right, systems, if there's no phosphorus, the roots will stop to grow. But in aeroponics, when you say the root system with -P, the roots continue to grow, and it didn't stop growing. One of the things we have that seed crops don't have is that we have vines, and also contain phosphorus and nitrogen. What happens during this time is that the phosphorus is actually translated from the vines to the developing roots. Ethiopia, what are the signals that the crop is ready? And in the next patch where it showed that the vines were already wilting, that it looked like it was nutrient deficiency. But what has happened is that the nutrients in the leaf being channeled to the roots because it was swelling. And if you didn't put out any fertilizer, the roots were trying to swell, we're using the nitrogen, the phosphorus in the leaves to be able to grow. So Mark, one reason why we didn't see the roots in -P is that it was just drawing the phosphorus from the so that's the one thing that the aeroponics system cannot do, is that to simulate mite deficiency. But other purpose screening for varieties, it can really be a method to screen for that as well. But yes. In other systems, it is quite cool that if you're doing multiple cuttings in your propagation block, that you're growing out the nutrients, and whereas the second cutting might have less potential for yield versus the first cutting, because you're expending the nutrients in the system. And who has the second cutting might actually have lower yield compared to the farmer who get the first cutting.
So any more comments? Our planting material specialist, how does the research of the role of phosphorus in storage roots come to what the farmer practice are? If you have any comments, do you think the gap there, or on the contrary, there's any?
Okay, thanks. I think my comment would be that it would be interesting to know the outcome of these, because you're working nowadays for a company. They got the shape, but it would be interesting to know whether they accept the same French fries were developed from this.
Sure.
Because of the several changes that
Right.
occur.
Yeah. Part of the process is they do quality controls. So they'll be able to tell if there's any metabolic changes or that shows up if you adjust the phosphorus. Yes. That's a very good point.
I can add on the presentation. Maybe something striking is the amount of time that most farmers do the 45 degree. I think the presentation, the flat horizon of landing is more beneficial. It's something that can
Good point. The noted in Ethiopia is that the temperatures were not very warm, not over 30. It was 27, 28. Have a very good chance of surviving because it will not dry up. So the moment it is in the ground, it will start to root. However, under conditions of heat, 30 or over, the topsoil can be a very hot place to be. And if you're planting your vine that way, you're effectively drying up the vine. So it only work, as you saw this failure, because they were upside down. they watered right away. And so you the initial transplant stress. In zone where it's not very hot, it worked well as we saw in Ethiopia. But a zone where it's hot and dry, probable planting might work better.
Maybe I am an issue of compaction. This falling level of combustion. People in our country they may have issues with environment and their erosion affected the problem. Mostly we use rainfall.
Mm-hmm.
But then it comes maybe in 1 2 months and there's a lot of flooding.
Right.
And then it becomes an issue. But we can look and see what's best to apply.
So it is.
This is kind of
Oh, comment. Okay. Yes. Right.
with a hard pan, just 3 kinds of deep.
I think the hard pan would actually benefit here because if you have a deep soil, your water would just go down. Whereas if you have a hard pan, you have the potential to catch water there. And if the root is able to grow to that area, then during very dry times, it actually has access to water. But if it's available, there's no hard pan, you effectively lose water to gravity.
Okay. And planting, the way we do the planting here, the horizontal planting can only be applicable for that type of farming. But when it comes to size, maybe in the containers that we use, more nodes inside the pot, then you know the more that are going to grow from you. And if your pot is small, even more roots will develop and there will be competition for the nutrients. So what you have to insert internodes inside the pot, they have to be very few. Maybe 2, 3 internodes can go in a container. But if you put horizontal more nodes, then you might end up with very, very tiny roots.
Very much, Arthur, fascinating presentation. To pick up on what you were asking, Mark, in terms of the implications for seed systems, I mean, I think there's things which immediately come to me. Just your comment on the raccoon crop for seed multipliers. I mean, this is a question that we've had in the community practice. Raccoons and what happens to the subsequent quality or vigor of the planting material. So it's definitely an area to follow up. And then I hope tomorrow, Bramwell, we can have more of a discussion because we're starting to use sand tonics for seed production. Bramwell is looking in particular at the nutrient composition on that. So particularly what is the role of potassium, the phosphorus and nitrogen. So I hope we can discuss that more tomorrow. And then just in our last community practice meeting, we started this whole discussion about seed production, not production, pots or beds, greenhouse conditions, and Kesseth, and again we'll look at this tomorrow, getting higher vine production using beds rather than pots. So there's a huge amount still to discuss. So thanks.
one last opportunity.
Okay.
Okay, thank you for the presentation. I would like to know how tubers or roots one is able to get when you plant using the flat method compared to the vertical method. And another thing did mention about weevils, that when you plant using the flat method, you minimize the damage of weevils to the root. How is that? Thank you.
Okay, the roots can be harvested from a vine that's planted flat versus vertical. The number is the same, but it's the sizing is different. Because in the States, they grade the roots when they harvest. So 500 to 600 grams will be your fresh market. You sell it in the store. The smaller ones will go to the canners or the French fries. The all fruits or the jumbos also go to the French fries. The fresh are the most valuable, and the farmer gets more profit from it. Planting it in a horizontal flat method, that means you have potentially all our fresh market size roots. So you have 10, you can get $20 per box. On the other hand, if you plant it vertically, some roots are going to be small. It's because they're from the nodes in the bottom. And some nodes are producing thin roots because when you plant it this way, the roots are very in direct competition with each other. Like you're actually manipulating the spacing so that less. And the distribution of the fertilizer and water is more even. So it's not the number, but the quality that's being changed.
When you plant horizontally, can you do that without irrigation system?
Well, the farmer, and you can do it, but if it's a different scale. But if it's for commercial, it has to be with drip because it's very hard to manage it with other irrigation means. Yes.
Okay. Anybody else? One last question. Well, time to thank you, Hatu, once more. So let's thank Hatu with a clap. Thank you much.
Thank you.